Now we're going to review the basics of the live Loretta projector, or LLP as we call it. This is a piece of software that converts surface EEG recordings into localization data internal to the brain in real time. It uses a technology known as Loretta, which is standardized low-resolution electromagnetic tomography. First, we're going to review a little bit about the EEG itself and then start looking at Loretta images. On the screen right now we have a 19-channel EEG which is scrolling at real-time speeds like a paper strip chart. And what we want to pay attention to is the distribution in time and the distribution in space of the different frequency components that we observe. For example, right here we just have an alpha burst and another brief alpha burst, primarily from the posterior. Here again, and here we have some theta activity along the top, which tends to be more frontal. Let me review the channels on this display so you can understand this. We have all the left-sided channels on the top of the screen, and we have the right-sided channels on the bottom of the screen, and we have the frontal electrodes at the top of each group, and then the posterior electrodes toward the bottom of each group. So for example, as we go through, there's a theta, frontal theta that you can see. That's actually eye activity. See how prominent that is. We have FP1, then F3, C3, P3, O1, F7, T3, T5, and then FZ. This is the left side, and then on the right we have FP2, F4, C4, P4, O2, F8, T4, T6, and then CZ and PZ. So for example, again, we see prominent bursts of alpha that are posterior. Here is some theta activity, which is primarily frontal. We also see beta activity, and it's not uncommon to see beta activity more prominent in the front. But what we're trying to pay attention to here is that as we see these distributions and fields of EEG activity, we'd like to know what's happening inside the brain to produce this type of activity. And Loretta is a technology which allows us to get beneath these types of waves and visualize how they would be appearing inside the brain. So we'll switch from this raw surface waveform screen to the Loretta live Loretta projection screen. And here we see a three-dimensional rendition of the head. In this case, we happen to be looking at alpha. And what we have then, if I show the scalp, you'll see we have the nose on the left, the eyes here. This is the front of the head. This region here is the back of the head. And as I rotate it, you'll see there is activity primarily in the rear of the head. And this is showing the alpha activity that we were seeing in the raw waveforms previously. So this particular display, which can be zoomed and altered in other ways, rotated, is showing the predominant alpha activity in the posterior. It's also showing that it's a little greater on the right side, as you can see in this representation. So this is the basic idea of using Loretta as a live EEG imaging system in order to image the voxel activity inside the brain in real time. The voxel activity that we're seeing is represented in units of current source density. Current source represents the amount of electrical displacement going on in the pyramidal cells, in the neurons themselves. So current source density is a valid measure of the amount of postsynaptic activation, polarization, and depolarization in individual locations. And with this display, we can either look at the whole brain, or we can zoom in and look at any voxels of specific interest can be looked at in detail as well. Now we're going to review the control panels that are provided with the live Loretta projector, explain how they're used and some of the principles behind them. We do have a lot of controllability, a lot of flexibility in the LLP. And we'll start from the top and then explain what some of the variables are. First of all, right now I'll note that we are looking at the whole brain, and we're looking at alpha activity, and we're looking at the activity from the rear right now. So we see what is actually posterior cingulate, as well as some parietal involvement in the alpha waves. We have the option of showing actual physical brain on top of this image. I'll go to the front to be able to show that, and I'll say I want to show the left brain and the right brain. And now you can see there's a physical brain superimposed on the image, which allows you to get a better idea of precisely which parts of the brain are involved in that activation. And some people like to leave this on, and some like to leave it off. And when we go to the surface potentials, you'll see why some people might want to use it on. That can be turned on and off. Also, the scalp, which is the third selection, can also be turned off. So with the scalp turned off, you're quite simply seeing only the voxels of the brain that are involved in the activity you're looking at. The third option is to show the electrodes, or the sensor positions. If we select Show Electrodes, we then have the sensor positions all shown in 3 dimensions in their proper geometric locations in relation to the brain. So for example, from this rear view, we see PZ is here, P3, P4, T5, T6, etc. So showing the electrodes is a very helpful, useful way of being able to refer the brain image to the sensor positions that were used to gather the surface data. Now the next selection is display, and we have several types of displays. Right now we're looking at voxels, which are the S Loretta projections. We can also switch to a scalp field, in which case we're now seeing the surface potentials on the scalp. And we can choose the smoothing method. This method is using the Laplace, and we can also choose spline as the smoothing method. And when you use spline as a smoothing method, then you're getting another type of very smooth estimation of the surface potentials. Another thing I'm going to jump down to show is that we have a damping factor down here on the bottom. And if you turn the damping factor down low, then the signals move very, very quickly. And as you move the damping factor up, they move more slowly because they're being more heavily damped. So that's a type of averaging that can be used. So again, with the scalp field, we could still show left brain and show right brain. So now we're showing the scalp field superimposed on an image of the brain. So you can now see how the scalp potential distributions relate to the underlying brain area. And we can show electrodes as well. So now we're looking at a scalp field potentials with the electrodes and with the underlying brain shown. So we recommend that you set these best use that you need for your clients' purposes at hand. So if I turn these off, we then will return to voxels. And I want to point out that you can choose the hemispheres when you show voxels. I can show either the right hemisphere, which I'm showing here. I can choose the left hemisphere, or I can choose both hemispheres for the voxel projection. Another series of choices that can be made for the voxels is the source. Right now we're looking at the acquired EEG, and we are looking at the alpha. So this is giving us the potentials of the in the Loretta of the alpha in the brain. We can switch though to z-scores, and now we can be imaging z-scores. And in this case, the z-scores are providing comparisons with a reference database. And in this case, the meaning of the scores changes slightly, so that a positive number means larger than the reference, and a smaller number means less than the reference. So we can show not only raw scores, but we can show z-scores referenced. We can also show EEG running averages, and this is another way of showing the average amplitude, the average energy over time. And we also have the EEG standard deviation, so now we can be imaging the variability. And the system will now be showing those regions which are the most variable, which have the largest amount of variation. And finally, we have Loretta Brodmann, which we'll talk about briefly. These are Brodmann activation patterns using the Skill-Kaiser algorithm for doing Brodmann imaging. We'll switch back then to acquired EEG, which is very typical because it's easily understood that this is the raw activity. I want to show some of the ability by changing the component being viewed. Right now we're showing alpha, and it's very clear that the alpha is maximal in the posterior region. This particular person does not have very much alpha in the front also, although occasionally we do see alpha activity in the frontal areas. What I'm going to show is if I switch to theta, we are now imaging the theta activity, and we should expect to see some differences in distribution. What we see, for example, is in addition to this posterior theta, we definitely see bursts of midline theta, and we also see asymmetric frontal theta. Right now we're looking down over the top of their head. As I rotate it, now we're looking at the back of their head, and then I'll rotate it again. Now we're moving to the front. And you'll see there's not very much theta, for example, here in this parieto-occipital area, but there is a significant amount of theta intermittently, particularly in this dorsolateral frontal areas. And now we're looking at the beta activity in the S. Loretta. And again, we see predominantly posterior in this case, although we see a little bit of anterior activity as we roll the head up and look over here. And we see a little more activation on the right. And as we then switch from beta to high beta, we see even more of this tendency where the high beta is more lateralized on the right-hand side. And we can rotate this brain to see where there might be other activity. We see relatively little in the front specifically. So that shows the basic ability to choose different components to view and image in real time. The next important capability that we're going to review is the ability to choose regions of interest, what are called ROIs. In this display right now, we're looking at the entire brain, and we're seeing that the regions where there's activation are going to be colored basically orange or red, and the areas that are not active are blue or dark. And right now we're looking at everything. And we have choices then. For example, the first choice is frontal lobe. So now I can look specifically at the frontal lobe and see the amount of this alpha activity which is present on the frontal lobe. I'll turn the electrodes on now to show it. So we can see that there's a small amount of activity which is actually a little bit posterior to CZ, still in the frontal area, and it appears to be primarily in that immediate postfrontal area. The next selection of a region of interest is the limbic lobe. So the limbic lobe allows us to visualize the limbic system and its components, and again image the amount of alpha activity specific to that lobe. And as we see, there's very little encroaching up on this posterior dorsal region. Primarily, there's very little involvement in alpha of the limbic system in this individual. The next region of interest is the occipital lobe, and here we're looking at the posterior, the occipital part of the brain, and then again we see that the predominant alpha activity, interestingly, is not so much in areas 17 and 18. It's not right under O1 and O2. It's actually a little bit superior, anterior to OZ, being more in the area of the posterior cingulate gyrus in this individual.